2 to 10 monosaccharide units. So, and as you can see in our last part of explanation, when two sugars join to each other, the joining style is from an oligosaccharide reaction, and therefore the link between two monosaccharide named as a glycoside and O-glycoside reaction. This type of reaction, this type of joint, glycoside joint, is highly resistant to alkali situation. However, acids can definitely dissociate this joint, this link, glycoside link is resistant, completely resistant to alkali. For example, NaOH, however, is completely sensitive to acids like HCl. You can see only disaccharide in your blood. Disaccharide, which means two monosaccharide. So you can't find any trisaccharide in your blood, tetrasaccharide in your blood, and etc. The only oligosaccharide that you can find in your blood are disaccharide, sugars with just two monosaccharides. What about the other oligosaccharides like trisaccharide, tetrasaccharide, and so on? Such type of oligosaccharide can be found in the cell and in attachment with other molecules. For example, you can find a trisaccharide with a protein. You can find a tetrasaccharide with a lipid, with a fat. So, such type of oligosaccharides are not free in blood, like disaccharides. Disaccharides are free and can be found in your blood, in your bloodstream. However, tetrasaccharide, trisaccharide, and so on could not be found in your blood in free form. They can be found in attachment with other molecules like proteins, like fats. These saccharides are mostly present in free form, as I said. And as I told you, those type with more than two sugars units are usually presented in combination with other substances like glycoproteins and glycolipids. What is the difference between glycoproteins and proteoglycans?
Doctor, the number of the protein is higher than the glycogen.
In which of them?
In the glycoprotein.
In the glycoprotein. Very good. And what about the glycolipid?
Glycolipid, the amount of the lipid is higher from the glycogen.
Very good. Yeah. So, this slide show you three important type of disaccharides that can be found in nature. The first one is maltose. Maltose. The sugar of Malta. Malt. Jo. So you can find maltose which which is used to produce beer, for example.
Beer.
Beer. ماء الشعير. This is a type of disaccharides, maltose. The other one, lactose. The sugar, which is found in milk. And the last one is sucrose. The sugar, which is found in such type of sugar.
عشان هو بس
فجر.
فجل
Wait a minute,
صاروخ ايش الكيلو؟ قلت ايش الكيلو؟ الله زين هاي استخدمناها الفجير ما ادري شنو هاي بصفه شنو؟
ايش قاعد يقول يعني قاعد يقول هذا مثلا موجود بهذا وهذا موجود بهذا.
ايه انا اقول لك الحليب موجود بالبقر والبقر موجود بالحليب.
اللاكتوز
اللاكتوز باثنين.
الحليب بالبقر.
حاشي شوف قول له بالحليب. لا هو اثنيناتهم
بفارسي شيء بفارسي.
ايش؟ English is sugar cane. I don't know.
بفارسي
I don't know. So this is the most
sugar cane.
zero type of
The
sugar
kid.
that you will always use. So these are three sugar cane, milk, and also malt. We have three types of disaccharides: maltose, lactose, and sucrose. So please let me know which the first one. What is the linkage type between these three types of sugars? The linkage type. The first question. As you can see here, in this type of molecule, the anomer carbon is in alpha form. So we have a alpha glycoside, alpha linkage. The next one is in beta form. So we have a beta linkage. What about the last one? Second question. Please tell me which of these three types of sugars are reducing sugar? Which of these three types of sugars is or are a reducing sugar?
سكروس.
First, what is a reducing sugar?
سكر.
First,
لا.
what is a reducing sugar?
من
Who can tell me what is a reducing
يسوون
sugar?
الشكر.
What is a reducing sugar? I explained it for you. A sugar with a free anomer carbon, which means free OH carbon, is a reducing sugar. So Which of this sugar is reducing one? For this, for asking this question, we should first know what is the anomer carbon. For the first one, what is the anomer carbon? Anomer carbon is carbon number 1. For this one, as you can see here, this sugar, which is glucose, have an anomer carbon which is not free, because the OH group is in the linkage with other sugar. But for this sugar, as you can see, the carbon number 1 is free. The OH is free, it's still free. And do not participate in a reaction. So, maltose is a reducing sugar, because this OH still can participate in reducing reactions with, for example, this OH. This OH, this inverse sugar cannot participate in reaction. However, this OH can. What about this one? Lactose. Same like maltose, because the first carbon is participating in the linkage. However, the next sugar has a free OH group. So this OH, therefore, both maltose and lactose are reducing sugars. What about the last one? Let me focus on this. Answer 2 questions. Is it a reducing substances? Why? Then why not? Please explain. Attention to the book and explain if you can. First question. What is the linkage between these two sugars? As you can see here, the first sugar is glucose, and the next one is fructose. This is fructose, and this is glucose. What is the linkage between these two sugars? What is the type of glycoside? Is it alpha or beta? Is it, as you can, remember, if we have an alpha sugar, so we have alpha linkage. For example, 2 glucose in maltose, two glucose attach to each other and produce maltose. 2 glucose. Carbon number 1 attached to carbon number 4. Alpha in an alpha form. Alpha 1 to 4. The next one. In lactose, carbon number 1 attached to carbon number 4 in a beta form. So we have beta 1 to 4. A galactose attached to a glucose. A galactose attached to a glucose. So you can find that the component of lactose are a galactose and a glucose. For maltose, we have 2 glucose. What about this one? What about sucrose? Well, the first sugar is glucose. The next one is, so please let me know, What is the anomer carbon for glucose? Carbon number we have 6 carbon in glucose. Which of these carbons is the anomer carbon?
one.
The first one, as you can see here. What about the fructose? Fructose is a ketose or aldose sugar.
Fructose,
Very good. So,
the ketone is the second.
second carbon. So, as you can see here, the second carbon of fructose is attached to the first carbon as in glucose. So, in both of them, the carbon which is involved in the linkage is a anomer carbon. So, the first one is in alpha form and the next one is beta. Therefore, we can name this linkage alpha or beta. We should say that alpha glucose linked to beta fructose. We can say alpha 1 to 2 or beta 2 to 1. We should say alpha to beta. We can't say an alpha or beta form. We have both forms of linkage in this type of sugar.
Doctor, can you explain it in the board?
Good. If we have a two sugar in with the alpha sugar number 2. And if we want to link these two sugars with each other, if an alpha sugar linked to a beta sugar, linked to another sugar, this type of linkage is alpha. If we have a beta sugar, beta, the linkage will be in beta. But if the first carbon and second sugar, in both of them, the linkage will happen in the anomer carbon. Both of them will participate in the linkage. So, in this situation, we don't have a beta or an alpha for linkage. We have both alpha and beta linkage. We have both alpha and beta linkage. For example, in maltose, the first sugar is in alpha form, therefore we have alpha linkage. In lactose, the first sugar is in beta form, the next one is not an anomeric carbon. The anomeric carbon is here at the first situation, so we have a beta form of linkage. However, In the last one, in sucrose, both carbon which is involved, which is participated in the reaction, are anomers. Therefore, we have alpha and beta, alpha 1 to beta 2, alpha 1 to beta 2 linkage. So, please let me know whether sucrose is reduced in sugar or not. Why?
There aren't any OH free.
There is no
Another-
OH free to participate.
And you?
Doctor, if there was two linkage, alpha and beta, there
Very
will
good.
be reducing.
All answers were correct because we don't have any free OH in anomer carbon. We have free OH, but those OH are not anomers on the carbon of anomer carbon. So, sucrose is not a reducing sugar. Sucrose is not a reducing sugar. Another examples. Please let me know whether maltose, lactose, and sucrose, whether raffinose is a reducing sugar or not.
Doctor, which of them?
Raffinose, let me magnify the slide. Do you think raffinose is reducing or not? Be silent, please. And just focus on the board. Is it a reducing sugar or not? Why?
Because the OH lactose is linked to the fructose. And
These two sugars. For the first one, for the first sugar, is it a reducing sugar or not? Because the anomer carbon is not free. The second one, is it a reducing sugar or not? Why? Again, we don't have anomer free carbon. And the last one, why? Is this OH work or not? Do this OH work
No.
for as a reducing substance or not?
No, it's fructose.
Very good. And in fructose,
This is the ketone.
so
It's second one.
the second carbon is the functional group, anomer group. So raffinose is not a reducing substance. But another example. Sucrose, lactose, maltose. What about isomaltose? Isomaltose produced from starch. Starch. Okay. Nişasta. Starch. We have two types of starch. Amylose, amylopectin. Then, when you use starch in your body, in your mouth, starch change to this type of molecule, isomaltose. Isomaltose. Please let me know whether isomaltose is a reducing sugar or not.
yes, it is
Why?
the functional group OH in the second.
Very good. And as you can see here, in the second sugar, you have a OH. You have a free OH. Therefore, isomaltose is a. What about cellulose? Cellulose. Cellulose produced from cellulose. Can anyone tell me when we or where we can find cellulose?
In the plants?
Very good. In plants, you can find cellulose in plants. The type of sugar, structural sugar in plant is cellulose. So cellulose can produce cellulose. Please let me know whether cellulose is reducing sugar or not.
It's reducing.
Very good. Because of this OH in the next sugar. So if anywhere we have an OH which is free in anomeric carbon, therefore we have a reducing sugar, and it is very important. Why? Maybe you have a patient that have a lactosuria, which means this type of patient cannot remain lactose in their body. And lactose exerted from urine or stool and other body fluids. Therefore, you can find whether in the urine, in other body fluids, if lactose exists or not. Which type of test can help you to this aim?
Urine test?
Oxidation.
What is the name of this test? Benedict test or Clini test. Very good. So you can find whether you have reducing substances or not. The name of test is Benedict test or Clini test or reducing substances test. Reducing substances test. So, what is the difference between cellulose and maltose?
Alpha and beta.
Alpha and beta.
The only difference between cellulose and maltose is the type of linkage.
Yeah, O and N.
There is no other differences between two types. But just for this difference, you can use cellulose as in your diet as a food, because there is no enzyme in your body which can dissociate, separate this type of linkage. However, we have alpha amylase. Alpha amylase in our body, which can extremely dissociate alpha linkage. We don't have any enzyme in our body who can dissociate, separate beta linkage between two glucose. Both of them are glucose. We have no enzyme in our body who can separate beta linkage between two glucose. We have just alpha one. Alpha enzyme named alpha amylase. It is located in our pancreas. And exerted in our intestine. When we use food, when we eat starch, for example in rice, in bread, when you eat bread, when you eat rice, those foods enter in your body, enter in your intestine, and their glucose dissociate, separate from the substances by alpha amylase. Alpha amylase, as I said, is located in your pancreas. It's a gland in your body, pancreas. So, there should be no or very low level of alpha amylase in your blood. If the level of alpha amylase rises in your blood, it means that your patient has a disease named pancreatitis. Pancreatitis. So you can find if your patient has a disease named pancreatitis with measuring alpha amylase in his or her blood. Such patients complain about a very hard and very serious cramp, abdominal cramp. They can't burden such pain in his body. It's very important to immediately manage such patient when they come to emergency department. Such patients suffer from abdominal pain, abdominal pain, ish or alam. Okay? Very serious pain in their body because of the inflammation of their pancreas, which named it pancreatitis. So, in pancreatitis, the level of alpha amylase, which is responsible for dissociation alpha linkage between two glucose, is extremely increased in your blood. Other example of disaccharides. This type of disaccharides made from sugar derivates. For example, we have hyaluronan or hyaluronic acid from sulfate, is from sulfate derivates of sugars, which means a sulfate group attached to sugar. For example, dermatan sulfate, hyaluronan, heparin, and chondroitin sulfate, tratan sulfate. These types of sugars are derivate sugars, which have an extra group, extra functional group. Most of the majority of functional group is SO3 or sulfate group. What is the property of sulfate group? As you can see here. What is the major property, the main property of
Oxygen?
No. It is a highly ionized charge. The charge of this group is negative. So this type of sugars are negative sugars, negative charge sugars, negative charge sugars. This type of sugars are negative charge. The last part of our class is polysaccharides. More than 100 monosaccharides. More than 100 monosaccharides. We have different type of polysaccharides. For example, linear or branched. We have amylase, amylopectin, and amylopectin. Amylose is a linear form of starch. Amylose is a linear form of starch. In contrast, amylopectin is a branched form of starch. We have two types of starch. And please consider in your mind that known of polysaccharides are reducing. We have no reducing polysaccharide sugar. We have no reducing polysaccharide sugars. We have structural storage polysaccharide. For example, cellulose is structural pectin is a structural sugar. However, glycogen and inulin are storage sugar. What is the property of inulin? Inulin is the storage form of sugar in potato. Homogeneous heteropolysaccharides. For example, polysaccharide, which is just consisted of sugars, and polysaccharides that are consistent of other molecules. For example, in this type, amino acid, proteins. So you have homopolysaccharides, which are just from sugars, and heteropolysaccharides from other sugars, two types of sugars. Let's give you some examples. Cellulose, ketene, both of these sugars are structural.
Structural and dissolved.
Which of them is structural and which of them is storage? Ketene or cellulose? Which of them is structural and which of them is
Cellulose structure.
Both of these sugars are structural. Where you can find ketene? In insects. Insects have ketene in their body. Insects, do you know what? Heteropolysaccharides. Heteropolysaccharides. For example, glycosaminoglycans. Glycosaminoglycans. Let me give you some examples of heteropolysaccharide. The last slide. Sorry. This one. As you can see here, in heteropolysaccharide like glycosaminoglycans, we have two types of sugars. For example, here we have glucuronic acid, which is attached to N-acetyl galactose. Two different types of sugar. However, in starch, you can only find sugar named glucose. You can only find glucose. In glycogen, you can only find glucose. No other sugar exists in glycogen. You have just one type of sugar. But for glycosaminoglycans, you have two types of sugars. For example, here, galactose plus N-acetyl galactosamine cratane sulfide, and so on. So, the summary of this cellulose is starch is glycogen. As you can see here, cellulose mainly found in plants. Amylose also found in plants, however, glycogen found in the body of animals. Which cells are in your body contain glycogen?
Tissue.
Which tissue or cell contains? The muscles and liver. Which of them has more glycogen?
Muscle.
Muscles.
If it's one cell, liver.
Very good. Subunit. Cellulose, as I explained to you, has beta in a beta form. However, in the start, you have just alpha form. In glycogen also you have alpha glucose. The type of linkage is 1 to 4, 1 to 4. Why? It mentioned here 1 to 6 for these sugars, amylopectin and glycogen.
Two functional group.
Was it branched?
Very good. In the branch, in the location of branch, you don't have alpha 1 to 4 linkage. You have alpha 1 to 6, which means carbon number 1 OH from carbon number 1 attached to an OH from carbon number 6. Very good. Very good. The branch. We have no branch in cellulose and amylose. However, we have per one linear, per 20 subunit, we have one branch for amylopectin, and per 10 subunits, we have one branch in glycogen, which means the branch number in glycogen is higher than amylopectin. The difference between amylopectin and glycogen is about their branch number. And the shape, these types are linear, and this one are branch. Any question?
Thank you.
Thank you for your attention.